Hey everyone, how you doing? Today we are going to look at DNA and how DNA is investigated, analyzed, and compared. Let's first go over the basics once again. DNA is a double-stranded structure with nucleotides in the center holding together two strands of DNA with the aid of hydrogen bonds. When untwisted, this double helix now looks like a ladder with those nitrogenous bases in the center and alternating sugars and phosphates. By the way, if you hear some noise in the background, it's my kid, probably on FaceTime or just walking around upstairs. All right, so now that we have the basics, I need you to remember that nitrogenous bases are really not these letters A's, T's, C's, and G's, but they are chemicals. And as chemicals, they have shapes. Now, what else? Well, let's take a closer look. You see these nitrogenous bases and their shapes. Again, those are simplified. These are just geometric patterns so that we could understand something much deeper. But what other chemical do we know of that has shape and because it has a shape can do a job? And that is enzymes. So I'm going to introduce to you an enzyme and I'm going to show you its shape. Now, where would this shape fit in this strand of DNA? And if we were to scan, we would notice, boom, right here. And enzymes have the ability to build things up, but also break them apart. In this case, it's going to break the DNA apart into pieces. Now, again, this is overly simplified, but it demonstrates a point. So let's practice just for a moment. We're focusing on CCGG, just like in this picture. It was CCGG created a unique shape that the enzyme fit into. So, and no, we're not going to change it. Oh, that's what I was looking for, this screen. In this strand of DNA, well, where would the enzyme break it? And what we're going to do is we're going to scan until we see CCGG. And just so happens we see it once again here. And the enzyme would cut it right here and here. And we can see how many fragments long this strand of DNA is. And it is 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. And this last one is 5. And we could do the same for these two also. But the fact is, we were able to do this, but you cannot measure DNA in this fashion. You have to use some tricks. And for that, I'm going to move to this website and show it to you. I'm showing it to you in this way because flash animation does not work on most devices that you all have. So let's go through this virtual lab together. And it says here you hold a plastic tube with some clear liquid in it. You've been told that the liquid contains DNA strands of several different lengths. Your job is to figure out what those lengths are. How will you do it? Well, it would be easy if it just happened that way, but it does not. So if the strands were as big as your shoelace, you could sort them by hand into groups and measure them. But DNA strands are molecules so tiny that you can't see them even under most microscopes. Is there a way to sort and measure the DNA strands in your tube even though you can't see or touch them? There is. It's called gel electrophoresis. Scientists use gel electrophoresis whenever they need to sort DNA strands according to length. This technique is also useful for separating other types of molecules, like proteins. How does this work? I love the sound effects. The gel is the filter that sorts the DNA strands. It's like a sponge made of jello with many small holes in it. See, this magnifying glass is now actually showing you that there are microscopic holes. So there are paths for which molecules can move from one end to the other. All right, those would be samples of DNA going into impressions in the gel. So we place DNA samples into holes at one end of the gel. Electrophoresis is how we push DNA strands through the gel filter. By adding an electrical current, we can make DNA move. You see, DNA has a negative charge, so it's going to move from this side to this side towards the positive. This is cool. You see, this is where the DNA originated from, and it's going through all those pores, and you're seeing it actually move. And which ones move the furthest? The shortest ones, of course. The shortest ones are the lightest, so they will travel further. So it says, short strands move through the holes in the gel more quickly than long strands. Over time, the shorter strands in the sample will move further away from the starting point than the longer strands. DNA strands of the same length will move at the same speed and end up grouped together. In this way, DNA strands in the sample sort themselves. Staining the sorted groups of DNA makes them visible to the naked eye. Although we can't see a single DNA strand, we can see large groups of stained DNA strands. These groups show up as bands in the gel. Now it's your turn to run the gel. The scientist's slang for gel electrophoresis.
Interesting.
Yes, it is. Here's what you need to make the gel. You need powdered agarose, that's the actual gel itself, buffer, a flask, a microwave, a gel mold, and gel comb. Making an electrophoresis gel is a lot like making Jello, but you don't want to eat this type. Ha 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 ha. Okay, put a small sample of agarose in the flask. Agarose is a dried powder similar to gelatin, but made from seaweed. So, uh-oh. There we go. Thought it wasn't going to let me do it. All right, add some liquid buffer to the flask. The buffer is a salt water solution that will let electrical charge flow through the gel. All right, I know what to do here. We're just going to heat it up. All right, we've removed the plastic wrap from the top of the flask. Pour the melted agarose mixture into the mold. Notice that the mold has tape on each end to hold the melted agarose. You don't want it to spill out. Now here's where the impressions that you saw earlier come from. Place the comb into one end. The notches in the gel mold are held in place. Again, these are going to make the impressions that the DNA will be put into later. Let the gel cool and solidify. This usually takes about a half an hour, but we'll speed up the clock for you. Carefully remove the comb, leaving empty wells for the DNA samples. Now it's time to set up the electrophoresis box. You need the gel you just made, an electrophoresis box, and another bottle of buffer. Pour the buffer into the electrophoresis box. Place the gel, still in the mold, into the electrophoresis box. We have removed the tape from the end of the gel mold. The gel should be just barely submerged in the buffer. The buffer conducts the electrical current from one end of the gel to the other. It will also keep the gel from drying out during the experiment. All right. You're ready to load the DNA samples into the gel. Here's what you'll need. Loading buffer, the tube of DNA, the DNA size standard, a micropipetter, the electrophoresis box containing buffer, and the gel and pipette tips. All right. With a clean pipette tip, use the pipetter to suck up some loading buffer. Then add it to the DNA sample. The sample's right here. The DNA samples are prepared in a clear liquid solution. That would be hard to see if you tried to load it directly into the well. The loading buffer contains a dye that makes the sample easy to see. It's also slightly goopy. This makes the DNA sample thicker so that it will drop into the well instead of floating away in the buffer. The DNA size standard already contains loaded buffer. All right, so we just took some of that loading buffer and put it into the DNA sample. Next, we will use the micropipetter to transfer the DNA sample from the tube into the well of the gel. First, suck up some of the DNA sample into the pipette tip. And we don't have to change this tip because it was already in both of these solutions. It's not going to become contaminated, so we will take some of that up. Next, there we go. Eject the DNA sample from the pipette into the first well. And now it is being loaded. The DNA has been loaded into the well. It says, nice job. In real life, loading the sample into the well will take some practice, so don't be disappointed if you miss your target the first few times. All right. Next, using a clean pipette tip, use the pipetter to suck up some DNA size standard. We got to talk about that standard in a little bit. It's kind of like a control. Transfer the DNA size standard into the next empty well. The DNA size standard contains DNA strands of known lengths. Running it on the gel will give you a reference by which to estimate the lengths of the DNA strands in your sample. Again, we'll talk about that more in a little bit. Load that up. Now to the electricity. It's time to turn on the electricity and run your gel. When you turn on the power, the black end will generate a negative charge. That's electrons. And red will generate a positive charge. Together, they will pass the current through the gel. DNA has a negative charge. To move the DNA through the gel, you must put the black cord, the negative charge, closest to the wells. Okay, so we got to plug this thing in. Plug the black cord from the electrophoresis box into the matching outlet on the power supply. Click the location. Okay, so we have to do this one, do that one. All right, and let's turn it on. Okay, your gel is off and running. Let's look into the gel box to see what's happening. Check for tiny air bubbles. Check for tiny air bubbles coming out of the electrodes at both ends of the electrophoresis box. These bubbles are your proof that the current is running. See, what's happening is that electricity is actually breaking water molecules into hydrogen and oxygen gas, so it will make bubbles. I'll take a look right here. Watch this. Watch this. They're moving. All right. Repelled by the negative charge, the DNA moves through the gel towards the positive charge. So it's actually attracted to the positive as well. On the other side, shorter DNA strands move through the holes in the gel more quickly than long strands. Over time, the shorter DNA strands will migrate further from the starting point than the long strands. We can't actually see the migrating DNA bands, but we can see the blue dye from the loading buffer as it migrates. You finished running your gel. We've taken the gel mold with the gel in it out of the electrophoresis box. The time has come to analyze your sample and estimate the lengths of the DNA. I'm kind of in the way here. Let me go right there. First, you need to stain the DNA in your gel using DNA staining solution. The stain is a chemical called ethidium bromide, which binds the DNA and shows up under fluorescent light. Although we can't see single DNA strands, we can see large groups of stained DNA strands. These groups will show up as bands in the gel. And you can see here a picture of the DNA, and it shows you where the ethidium bromide is actually going to bind. Let me now go back over there. Drag the gel out of the mold and put it into the DNA staining solution. But because ethidium bromide binds the DNA, it can damage the DNA in your cells. If you stain a gel in real life, wear gloves and avoid direct contact with the staining solution. Again, love the sound effects. It takes about a half hour to stain the DNA in the gel. We'll speed up the clock. Remove the gel from the staining solution and place it on the UV light box. Turn on the box. All right. Here is your standard size. So this becomes a measuring stick, a ruler, let's call it, to now gauge how big the bases are in the sample that you're interested in. So if this is 6,000 base pairs long, that's 5, 4, 3, 2, and 1,000. Where does this one line up within? And it's asking us to put in a number. Of course, pause and try it for yourself. If this is 4,000, that's 3,000. Where does this lie? And if this is 2,000, that's 1,000. Where does this band lie? So these are our estimates for how large these base pairs are. All of your estimations are correct. The estimated length in base pairs of each band of your DNA sample is based on how they line up with the bands of the known length in the DNA standard size standard. The top band is approximately 6,000 base pairs. The second is approximately, I put down 3,400 because I didn't think it was exactly in the middle, but they're saying 3,500 is a good estimate. And the third is approximately 1,500 base pairs. So congratulations, we've done it. And let's go back to the slides that I have prepared. So let's summarize in a couple of ways. We start with a DNA sample. This is what the DNA would look like if you could actually really see it. But we need to cut it into pieces using enzymes called restriction enzymes. They cleave, meaning cut DNA into smaller segments of various sizes because, well, the code of the DNA is going to vary as you go down the length. So it's going to find multiple locations. Okay, so here are the different sizes of DNA, and you load it into the electrophoresis chamber, into the gel. Here, the DNA segments are loaded into the wells in a porous gel. The gel floats in a buffer solution within the chamber between electrodes. The electrical current passes through the DNA, through the chamber, taking the DNA with it towards the positive side because DNA itself is negative, and the smaller segments move faster and further than the others. This is another way if you prefer seeing it this way. Enzymes are used to cut the DNA at specific locations. The enzymes, restriction enzymes, find the locations due to the DNA's code. The DNA fragments are placed in a specific type of gel inside an electrophoresis chamber that has electricity attached to it. The DNA fragments have a negative charge and are attracted to the positive side and repelled by the negative side as well. The electricity is turned on. The smaller fragments will move further closer to the positive charge than the larger ones. Where the fragments come to rest, a visible band is formed. That is, if we stain it, the pattern bands are similar in many ways within a species. So I would have a similar banding pattern as you, but unique to the individuals. We're going to see some little differences between us. This is used for analysis similar to the way fingerprints are used. Now over here, this is just a simplified picture. Actually, wait, this is an actual gel electrophoresis picture. On this right side, you will see the standard with the known base pairs, how long they are, so that you can measure how long these are and then compare them against each other. Over here, it's the same thing really. It's just drawn a little bit more clearly. Here's another picture, but here's something else you can do. You don't need the standard. You can just compare one to the other just to look at similarities between them. I should say, the different samples. So for instance, bear 1, bear 2, and bear 3. In this example, this was given on a Regents, and they asked you which two bears are more likely to be more closely related. So why don't you do that? And, well, that's gel electrophoresis for you. Have a great day.